I am René Williams. I am assistant professor at the Molecular Photonics Group at the University of Amsterdam. And today we will discuss translational and rotational molecular motors. We will talk about the work of Sauvage, Stoddart, and Feringa, the Nobel Prize winners of 2016. There are many molecular motor molecules in action in your body at the moment. Think about kinesin and HPAs. These biological motors are a big source of inspiration and have led to rotaxanes and rotational motors. Today, we will discuss molecular motors, rotaxanes, molecular motors of Feringa, and they can be activated with electrochemistry, but also with light. The rotaxanes, but also the Feringa motors, they are light operated. Here we have a representation of a rotaxane. We have a stick, we have a station in yellow and a station in orange, and a ring that is over it through a non-covalent bond is in blue. Now, if we excite this naphthalimide, we get to the excited state, and if we do a trick with electron transfer, then the affinity for the blue ring becomes higher. This state is now lower in energy, the ring will move. The rotaxane will shuttle, and the ring will end up at a different station. If we lose the charge, this situation is less favorable, so the ring will move back. It will move back to the orange station again. This is a cartoon you have seen in supramolecular chemistry and nanomaterials. We often use cartoons to represent systems. The rotaxane, the stick and the ring, we have a power stroke and we have a recovery stroke. So the ring moves up and down. They call it a molecular piston, a molecular machine. So this is the structure, a naphthalimide, a chain, and at the other side of the chain, there's the succinimide station. And it works through hydrogen bonds between the red ring and the stations. So hydrogen bonding is the binding. In normal conditions, it's mainly at this side. So the ring is at the blue station. Okay, we either use electrochemistry or we shine it with light. If you shine light, this naphthalimide, it likes to get an extra electron. It's a good electron acceptor in the excited state. So you get to the radical anion. It has a higher affinity for the ring. So the ring is more likely to be here. If the naphthalimide loses its electron, if the electron goes back to this D plus point, the oxidized donor, then you get here, and now it moves back. So in this way, you can operate a rotaxane, you can make it shuttle with pulses of light. If you think about the energetics, then in the ground state, this situation will be slightly lower in energy. This one is slightly higher, and in between, the energy of the state is higher, but it can go over it with thermal energy. So then we do a trick. We zap it with light, we put an extra electron on it, and thereby we change the energy landscape. You know that the energetics of reactions in photochemistry, it's the same. And you go to the lowest state, and that's often different in the ground state if you compare it to light-induced states. By putting an extra electron on this side of the molecule, this well becomes lower, and that's why you end up there. In between, the ring is diffusing. The ring is going up and down all the time. Depending on the state of the station, it likes to stay at one side or it likes to stay at the other side. So in that way, the shuttling occurs. And so now we are at this state because it's lower in energy. When the ring basically is on the thread, you are in a high energy state and it uses just thermal energy. So if you would freeze it, then it wouldn't happen. If you have room temperature, it can go to this high energy state. It uses the thermal energy for the translation. That also happens in natural motors, actually. We use light to shuttle a ring over a thread. It goes up and down. What do you know about photochemistry? With photochemistry, we have a ground state, and we have absorption, and we have an excited state. The light has to be absorbed. If there's no light absorption, nothing will happen. In this case, this is just the absorption spectrum, very simple absorption spectrum that you also recorded maybe in your first year practicum of the naphthalimide. If you use 340 nanometers, it could work. If you use 600, it will not work. So you have to have absorption. That's logical. Now, if you make an excited state, if you check your banknotes, if they are real or not, you put them under a UV lamp and you look at the light that comes off. This is emission. the molecules, they also show some emission. So if you have the absorption event, this state, it can emit, it can show fluorescence. It gives you a tool to examine the excited states. So in this case, you can think about the emission of this naphthol image. You have all seen emission, fluorescence. You've all seen that. Checking the band notes or these stars at the ceiling of the children's room. But next to looking at emission, you can also look at absorption of the excited state. So by light absorption, you make an excited state. And that state in itself, it also has an absorption. And here is somebody basically doing that, demonstrating that. So we call it transient. So transient just means that it passes away. It's not permanent, but it's transient. And it's absorption spectroscopy. So here we zap the molecules with 355. They absorb the light. We just saw that. We saw the absorption spectrum, so we know it happens. And that is depicted here. So here you see the laser that hits the sample. Now we create the excited state here, the blue line. But this state, it can absorb. So if you have clever tricks, you can use here a white light beam to measure the absorption right after the laser hit it. So in that way, you can monitor the absorption of the excited state. And for these rotaxanes, if you shuttle them with light, everything happens in the excited state, or the important things happen. And so you need to probe it. You need to prove it. So this is a typical absorption spectrum. It's actually a differential absorption spectrum. So it's delta A of this particular naphthol emit state. So that brings us to Jablonski diagram. Has anybody ever heard of the Jablonski diagram? So this is the typical Jablonski diagram. It's a state diagram. If you think about molecules and materials and light, you always have energies of the states. So here we have we call it the S0. Okay, normal molecules have a singlet ground state. It can also be different for metal complexes or other materials, but most of them, they have a singlet ground state. So if you excite it, you get into a singlet excited state, which can be S2 or S1. If you go to the S2, then you have some heat, then you have internal conversion. It goes from the S2 to the S1. Then you can have vibrational relaxation. You go to the lowest level of the S1. So you go to this level. There you can have, for instance, reactions, but you can also have triplet state formation. So what is the difference between a singlet and a triplet? Do you know? Do you know what's the difference between a singlet and a triplet? A singlet excited state and a triplet excited state? We were doing electronic spectroscopy. We're exciting the molecules and we're getting electronic transitions. So normally you have a spin can have alpha or beta spin. So it's up or down relative to magnetic field, for instance. If you have a singlet ground state, one is down, one is up. If you have a singlet excited state, one just goes up. So it's still a singlet. But now if one flips, you have a triplet. So triplet has two electrons with electron spin that are in the same direction. It has a fundamentally different character. Does anybody know a molecule that has a triplet in the ground state? Oxygen. Oxygen has a triplet ground state because of the MO diagram, because of quantum chemistry. Oxygen is a triplet ground state. That's the reason why we're alive. If it would be a singlet ground state, we couldn't use it. Not reactive enough. We can have intersystem crossing, spin orbit coupling, and we go to the triplet. The triplet can show phosphorescence. That's this long-lived emission. Short-lived emission is usually from the singlet. We call it fluorescence. Normally, if you excite to a higher state, you end up in the S1, and from there everything happens. It was discovered by Kasia and Vavilov. This rule is named after them. And there's some time scales. So these processes are usually faster than picoseconds. Fluorescence is usually in the nanosecond region, and phosphorescence can be microseconds up to seconds. This is the basis. With this, we can detect excited states and the character. So here we have all the states. Here we have the excitation. So this is the excitation of our rotaxane. We end up in the S1. We can monitor the fluorescence. We can monitor the absorption of this short-lived excited state, transient state. We can also measure the absorption of the triplets. Now, the triplet space plays an important role here in this rotaxane business. So the transient absorption of the triplet state, it is nanoseconds to milliseconds. So in this way, you have to think about this molecule and the shuttling up and down with the light. The work on these rotaxanes, it used this technique to show that it actually works. So they look at the absorption of the excited state. Now, at very short time, you see this is the triplet. Now, at a longer time, this is the peak here is the radical anion of the naphthal imide, the Ni. But it's this unit here. This is a typical absorption spectrum. You can check it from other sources, literature. And you see that during the measurement, this peak, it shifts. It shifts to the blue side. And okay, they have proven that that peak, the shifted peak, belongs to the hydrogen bonded naphthalimide that is hydrogen bonded to the ring. So the spectrum of this species, of this unit, it's different than this one. And that change, the change of that in time, that tells you the shuttling and how fast it is occurring. These people, they used a rod with a ring, they zap an electron with light, they use light to put an electron on one station, then it likes to stick there, and they monitor the absorption of that state in time to show how the process occurs. So they use transient absorption to monitor the shift. This is the starting state. Now you have the naphthalimide that is not coordinated by the ring. And here, at the other side, you have the spectrum that belongs to this species. They measure the same process with time-resolved infrared. So they have different ways of showing it, showing that the ring shuttles over the axle or the rod. If there's no ring, it doesn't shift. If there's this ring, they have the shuttling. And so the shift of this peak, that gives the rotaxane shuttling rate. So it's in a microsecond. The ring goes from one side to the other in a microsecond time scale. So that is shuttling of rotaxanes. Can we understand that shift? Then you can say that the ground state here is more stabilized. So the energy difference becomes larger. It starts at 415 and it shifts to 411. And so there's a shift in this band. We can sort of understand it. We have clever molecules. We have motor molecules. We have to show that the ring goes up and down over the thread, that it can be repeated, that it has a certain rate, that the rate maybe depends on the conditions. So here we see some properties. The absorption spectrum in blue, this is the absorption of this unit. That's the chromophore. The other units, they absorb, well, mainly below 300. And this is the emission. The emission and the absorption have a nice symmetry. That's a typical what you see in photochemistry. This here is the overlap here is the 0,0 energy. So that's the energy that it can use. We have absorption. We go to the excited state. We can monitor the fluorescence. We can see what changes there. To make the ring have more affinity for one station, we pump an electron on it. We use the excited state and electron transfer to put extra electron densities on these CO units. That is how they did it. This here, it is DAPCO, diaza bicyclooctane. It's very rich in electrons. It's easy to oxidize. It's easy to give up an electron. And so in this way, they were able to create this state where there's an electron on the naphthalimide, and this one here is oxidized. This is from the singlet, but the singlet actually is short-lived. It's too short-lived to do the shuttling. They used a triplet. They used a triplet excited state because triplets are intrinsically more long-lived. They used a triplet and the same electron transfer to make a similar state that has triplet character. So the triplet charge transfer state, we can have triplets for normal local states, but also for charged states. So the triplet lives longer, and that can do the shuttling process. It can also, the ions can in principle also diffuse, and that's also what happens. The oxidized DAPCO, it just diffuses away, and after a certain time, it diffuses back. So that's why it recombines. So in this way, we have 22% yield of this state that can do the shuttling. The rest actually is lost. So it's not 100% efficient, but it does work. If there's two species in solution and they have to collide to do the reaction, what do you call that? SN1 and SN2, nucleophilic substitution. The 1 and the 2, what do they mean? For an SN1, you have dissociation, you have the carbocation formation, which is stable, so it can react with anything. But if there's an attack and something has to leave, that's an SN2 reaction, so it's second order. This is also second order decay kinetics. If you monitor the concentration in time of this recombining step, where the electron goes back, it's also second order, so it's not simple. Second order kinetics. And so the important thing is, they use this naphthalimide, and it has a very fast triplet state formation, and they use that triplet. If they would have used the singlet, if they would have focused on the singlet, they would never have shown the shuttling. So you need to have a long-lived state, in this case, a triplet charge transfer state. And although it's only 20%, but you can monitor all the steps very well with all kinds of techniques. You can see that also in this rotaxane business, spin state, it is important. And this is the molecule, zapping it with light, you have the electron transfer, you have the oxidized DAPCO. You create an anion here. This has a higher affinity for the ring, better hydrogen bonding. So the ring moves there, it sticks there, it likes to be there. And then you have recombination. The electron on the naphthalimide goes back to this one. You create the neutral state of the donor. The ring is now in a situation that's energetically unfavorable, so it moves back. So in this way, you can make a cycle, you can induce it with light, you use electron transfer, and you use the spin state. So you use the triplets. People in England and also in Italy, they did the shuttling with electrochemistry. So then you have to tune your apparatus to a certain potential, and then you can see one state. You have to tune your apparatus to another potential to get the other state. It also works. It's a similar principle, but doing it with light is very elegant because it is an external trigger. So in that way, people have shown shuttling of rotaxanes. Well, there's a link of these rotaxanes to NMR and the time scale. And what I just showed you, the emission, we said it's on picosecond time scale. So 10 times 10 to the minus 12 seconds, very short. The shuttling was a microsecond time scale, so that's 10 to the minus 6 seconds. Pretty short, I would say. So you are chemist, you probably have used NMR. Well, can you tell me the time scale of NMR? Or how, well, what do you estimate? How could we explain it, use it? What is it? What are important factors there? Yeah, millisecond, exactly. For people that do laser spectroscopy, that is pretty slow. But how can we explain it? How can we explain the time scale of NMR? Well, we can use a rotaxane system to explain it. This is, again, a rotaxane. It's quite a short one. So you see here is the thread. Here's the ring. Depending on the temperature, the ring, it pirouettes, it rotates around the thread. So we can use this as a tool to monitor processes. You can see that it also, the ring in blue, it has a certain shape. Somebody has written something here on the hydrogen atom. What is this shape of the ring? What does it make you think of? Is there a very simple molecule that you have seen in your first year of chemistry that actually has a very similar conformation? A chair-like cyclohexane. And if you look here, in this way, it's a chair-like cyclohexane, and it has axial and equatorial protons. These are equatorial and axial protons of cyclohexane. You have to be able to draw such a molecule with all the substituents with the equatorial and the axial positions. Now, what happens if we flip this ring? If we flip the cyclohexane ring, the axials become equatorial and the equatorial become axial. And you can draw it yourself. So we can monitor this with NMR. So if we go to low temperature, we basically stop the pirouetting and we have one conformation of the ring. So we can see separately equatorial and axial protons. It is two separate signals. So if you start heating, you have something you call coalescence. So the peaks, they move together and they become one peak. Here they are coalesced. They are together now. So now the NMR cannot make a distinguish anymore because it's too fast. The coalescence, it happens, and the shift here is about 1 ppm. And we have a 500 megahertz machine. And that gives us 500 hertz, 2 milliseconds. The time scale that you can determine with NMR, it depends on the ppm difference that you start with. If it's 10 ppm, well, you have a factor 10 more, and how good your spectrometer is. So you have a 900 megahertz machine, then it's also like a factor 2 faster. That gives you the time scale of NMR and a rotaxane that is used as a tool. The pirouetting of the ring of the rotaxane, you can use it for that. And you can study the movement of the ring. The people here in Amsterdam, especially Woutersen, he was very important here. The pirouetting of the ring or the shuttling of the ring, you can make it faster if you add water. So water is a lubricant for molecular machines. That sounds really clever. And here they show that. So of course, water, it competes with the hydrogen bonds. And that's why the ring is fixed and not so tight. It goes up and down more fast. And here you see this is actually exactly the same molecule. This one and that one is the same. So you can understand that this pirouetting is also influenced by water. So water is a lubricant for molecular machines, rotaxanes, hydrogen bonded rotaxanes. And that people have showed it with these types of experiments. So this is all rotaxanes, and rotaxanes are molecular machines. It is first at one station, then it goes to the naphthalimide, and then it goes back. And this is the charging. So now we put an electron here. The reset is the charge recombination. So the ring is less bound. So it moves easier because it can escape easier. If you use too much water, then you will get a problem. And I can tell you, you see this here is 2013. The first work was 2001. That was the first science paper from Amsterdam on this topic. And that guy, Schurz, I was in the PhD committee. Schurz already knew that if your acetonitrile is not dry, then you have problems. He knew that he has to have very, very dry acetonitrile. Otherwise, it becomes too fast. If there's too much water, you cannot show the shuttlings that easily. We already knew in 2001 that water played an influence, but here they used that influence very nicely. It's been done with time-resolved infrared. So you can monitor the CO regions. So you can monitor the process with a different spectroscopy. So these are the papers. This is the first one. This shows the operational mechanism, this is the lubrication. So that is the rotaxane work from Amsterdam with David Leigh. It is related to the work of Stoddart. And Stoddart got the Nobel Prize for his rotaxane work. So this here is Sir Fraser Stoddard. I met him in Vienna one time. He's a very, very nice guy. He has a good sense of humor. But these are the molecules that he worked on. And you can see it is a similar system, but it has more stations. So it is also a thread with different groups attached to it on the thread, and a ring. And the ring, it can go from station to station. It can move. So here we have the stoppers. If you don't have a stopper, then the ring will move up. And you see that the stations, they have different atoms, different groups in it. So here you see some sulfur atoms. Here you see some positive charges. This is naphthalene dioxy, so 2 oxygens on a naphthalene. It's relatively electron rich. This is relatively electron poor. And this is a good electron donor, but it can also be easily oxidized. They all do stuff with light or with electrochemistry. If you're interested in the work of Fraser Stoddard, I can recommend these 2 lectures. This is before he got his prize. He is very relaxed, very entertaining. This is his actual Nobel lecture. If you watch them both, you can see how nervous he was. It's really, really interesting. Excellent work of a Nobel Prize winner. So these are the typical molecules that he worked with. So here you see again, this is a big pyridinium with 2 plus charges. It is a dication. It's a good electron acceptor. It is electron deficient. It likes to take up an extra electron. This here is a tetratiafulvalene. It's a very electron-rich unit. It's double bonds with sulfurs on it. It is very easy to oxidize, and this is like in between. Here you have a catenane, which is redox active. So this is the molecule. This is the cartoon. The cartoon is just two rings. Here, this is the viologen, the bipyridinium, and the red is that one, the green is that one. With there's now one electron is taken out. This here is the unit that's most easily oxidized. So it's a plus point now. You take out from a neutral molecule, you take out one electron, so it becomes positive. There's one electron remaining. So it's a radical cation. It's a radical cation that is formed depending on the state. Now here, this electron-rich unit, it likes to be in between two electron-poor units. There's like a charge transfer interaction. The pi-pi interaction is like extra. It is helped by the transport of charge. So if you oxidize this one, it doesn't like to be there anymore. It moves out, and its position is taken over by the red part, which is like intermediates. So if you put back the electron, if you convert this radical cation into a neutral state, then it flips back. It's a catenane. In a way, similar processes as we showed before are used. You use electrochemistry to take one electron out or put it back. We used light to put one electron on and remove it again. So in that way, you can control the position of the groups on a catenane by using electrochemistry. So you use the redox. Well, it's very easy to oxidize. It's a very electron-rich unit. If you have a double bond with amino groups, that's also very electron-rich. And these units, the blue units, they are very electron-poor. Electron-poor units, they like to interact with electron-rich units because then you get charge transfer interaction, even in the ground state. So this one is neutral. It's more difficult to oxidize, but it's still relatively electron-rich. Especially this ring, that is typical for the work of Stoddart. Almost all his papers contain this unit. And you have rotaxanes, catenanes that can move with electrochemistry. It's also electrochromic, so you can change the color of the system depending on the oxidation state. And so you can go from blue to green to red, so it's an RBG system. Maybe you can do something with that. So the typical materials of Fraser Stoddard, this pi acceptor and pi donors. So here you can see how you make this ring. So it's basically the nitrogen that's attached here on the bromo. It takes it out. You can make the ring of Stoddard. You can template it with a crown ether. If you do that in the presence, you have already the pi-pi interaction between electron-poor, electron-rich. And that way you can make the catenation and close the bond, and then you have it. So here you have the non-covalently linked rings, and it's like a signature for the work of Stoddard. He incorporated similar principles into rotaxanes, molecular switches, molecular railways. So here we have a system with two identical stations. So basically the ring can be on this side or on the other side. Of course, it's difficult to prove this. You have again, here is a stopper molecule. Here you have a methoxybenzene, and here you have this typical Stoddard ring, bipyridinium, the metal viologen, they call it. So a very electron-poor ring. And that can move over the axle. The work of Fraser Stoddard, it contains all these kinds of elements, and it uses it in all kinds of rotaxane-type systems. Here's another example. So here we have again this ring on the TTF, this sulfur molecule, and it says ring goes here unless TTF is oxidized or viologen is reduced. So that is the hint of how the system works. Ring goes here when viologen is reduced. So when this is reduced, the ring moves there. And the ring goes there when TTF is oxidized. So if you take an electron out of this one, then this ring moves there. So in this way, very complex molecules with different stations on the thread, and you can control the position of the ring by playing with the conditions. We begin here where the ring is on the TTF molecule. That's this one here. You see in green, you see these 5-membered rings. So the ring is on the TTF. Now, if we reduce it, it moves to this one. So both the units are actually reduced. If we oxidize this one, it ends up at the red station. So you oxidize the TTF. It doesn't like to be in this ring anymore, and the ring ends up at the naphthal station. So it's a tristable, and here you actually have radical pairing. So they are both radicals. The ring and the thread have a radical state. And that's again something novel for rotaxane systems. And so here you see that both units are radicals and they're sticking together. And so we have the stoppers, we have the naphthal oxy station, and you have the naphthalimide station here. After reduction, these two units like to stick together while they're actually both reduced. So that shows you the complexity of the systems of Stoddart. And one little more example. Here you see that Nobel Prize winning work on rotaxanes is combined with the recent Nobel Prize on click chemistry. Here, these rotaxanes were made with the click reaction. In 1999, I was working in New York, and at that time, Stoddart was doing stuff with Hewlett-Packard. They made switches. So they had metal bars, in between they had the rotaxanes, and they could switch the position of the ring and thereby change the connectivity between the two metal bars. So he made the front page of the New York Times in 1999. So here you see such a rotaxane system. This is the typical Stoddart ring, the naphthal station, this TTF station. Here a little bit more extensive molecules. So they put it in between two metal bars and they were able to switch it. So they would switch the position of the ring with the voltage. So it is very nice to show that you can change conduction between metal bars by switching the position on a rotaxane. In practice, I have to admit, it's too slow. You cannot make a computer out of it because it's too slow. There's another guy that also got the Nobel Prize that it was shared with 3 people, and that is Sauvage, Jean-Piere Sauvage, chemical topology. That is important here. So this represents one of the important contributions of Sauvage, complex rotaxane-like molecules. Here you see an X-ray structure of 2 molecules, depicted in black and depicted in gray, self-complementary. This is the ring, this is the stick, and it self-complements, hermaphrodite molecule. On to the work of Feringa. So Feringa, he used double bonds. It is light-induced steps and thermal steps that make the molecular rotor go around. This is another link to a Nobel lecture. So this is the lecture of Feringa, which is also highly recommended. Have a look at it. Ben uses chirality. So if you look carefully, this carbon here, it's chiral. So he uses intrinsic chirality of his molecules to make them unidirectional. Without the chirality, they could still be rotational, but they probably will not be directional. So they're unidirectional. His systems, they move in one direction, and that makes it really, really unique. He uses a photochemical carbon-carbon double bond isomerization. So he uses Cis-Trans isomerization, or ZE, as people also call it. They're relatively small molecules, but they're complex, and they contain chiral centers, sometimes even more than one. So this is the basis of the work of Feringa, and this is why he got the Nobel Prize. How does the bond flip? So here you see this bond with photochemical and thermal heating steps. It isomerizes. It isomerizes all the time. So that brings us to, how does a bond flip? Can anybody tell me what is important in this bond flipping? If I want to flip this bond, what has to happen? And what are the important states in between? What happens if we use light? So we have excitation of a double bond. We have a pi bond, pi orbital. We put one electron in the pi star. It has a different orbital symmetry. What happens if you put an electron there? Do you increase the bond order between the carbons? No, you decrease it. You decrease the bond order. What's the consequence? It rotates. Why does it rotate? Who invented that it should rotate? Mulliken has stated a very long time ago, the energy of excited single state is minimized when the two p orbitals are orthogonal. So that means we start off here. This is the ground state. We have a filled pi orbital. The pi star is empty. Now we excite it. It's photochemistry. We put one electron in this pi star orbital. Symmetry is different. Now Mulliken tells us energy is lower if they are orthogonal. So you go to a state. We're going to a twisted state. A twisted state in which the pi orbitals are orthogonal. So that's represented here. Now people have studied this state a lot. They call it a phantom state because it's very short-lived. And if you go in the singlet state, it's usually called a zwitterionic. What does zwitterionic mean? What does it mean, zwitterionic? A plus and a negative charge. So it's like drawn here. They're basically isoenergetic in this case. So this represents a zwitterionic phantom state, which is the twisted state, according to Mulliken, where you end up. So here we start, we excite, we have part of the rotation, and we're now here. So here we can go from the S1, we can have conversion, internal conversion to the S0, to a high level of the S0, and we get here. So here we can have branching. If it's totally symmetric, then we can go to the trans. So the isomerization from the cis to the trans, in a photochemical way, you can describe it with this scheme: excitation, this orthogonal state, and then you end up here. So basically the branching here, it tells you about the quantum yield. There are also tricks that you can do. So you can start with a triplet. You can use triplet sensitizers. So if you do that, we somehow excite it in a different way. We end up here. So now, and we start again with a ground state, 2 electrons in the pi orbital. So we end up in the triplet state. So these 2 electrons have the same direction. It's a triplet excited state. We can still have isomerization. We go again to a orthogonal state where the pi orbitals are orthogonal. Now people have found out that the orthogonal state, if you start from a triplet, it usually has biradical character. Generally, that's true. Of course, here you can also, either you can go that way or that way. So you, with the intersystem crossing, you can go to the trans or to the cis. So this represents the photochemistry, the orbitals, and the spin of a simple isomerization for the simplest molecule you can imagine. And it shows you a zwitterionic phantom state and a biradical phantom state of Feringa motors. Processes that are happening in our eyes, they're also Cis-Trans isomerization. So vision is also very important. So here we have the same, it's not the Feringa motor. So here we have the cis-alkene, we zap it with light and we get the trans. This is happening at the moment in your eyes. It's the same process as the process of Feringa motors. And it's happening all the time. We go from a cis-alkene to a trans-alkene. Of course, it's a very complex, long molecule. The Feringa motors, they use internal chirality for the rotation, for the uni-directional rotation. The eye, it uses external chirality because the retinol, it is inside a protein which has a chiral nature. The pocket and the chirality is important for the efficiency of the isomerization in your eye. And so here we have the light-induced step. We go from cis to trans. It actually then is leaving its cleaves. It has to be regenerated. And so here we have the cycle. We have a picosecond step. Then it's batho, rhodopsin, lumi-rhodopsin, 20 microsecond, 500 microsecond, and there's even a 60-second step in this whole cycle where it is then reformed. This state is reformed. It's a chemical reaction. It's the shift-base formation. That's all happening in your eye, the isomerization, and the whole biological cycles. It works very nice, but the motors of Feringa, they are, of course, maybe more practical for application, for making products. Now, people have, especially Mr. Matisse, he has measured this isomerization in the eye. It happens extremely fast, 200 femtoseconds. So 200 times 10 to the minus 15 seconds. That's time scale. That's how fast it happens. He measured that with techniques, transient absorption, for instance. So here you see some kinetic trace. This is time, this is signal. You see that within 1 picosecond, everything is finished. If you look with a faster time scale, in about 200 femtoseconds, so we are here. Here, most of the things have happened. The measurements show with a 35 femtosecond resolution that in about 200 femtoseconds, everything has happened. So the whole process is extremely fast in your eye. This is rhodopsin. You can think of it, we just saw the potential energy surface of a simple alkene. This is how you can express it for retinol. So here we are at the ground state. We have vibrational wave packets. We transport it to the excited state. We excite it. And then within 200 femtoseconds, we are directly crossing to the other state, so to product state. So this process is happening in your eye extremely fast. And that's why it works. If it would be slower, then we would have problems. And the message is this protein environment is also a chiral pocket, and that can give you a link to the internal chirality of Feringa. And so basically, if you make the potential energy surface asymmetric, then your yield will be larger because you're not ending up at this top, but you're ending up here. So then you can have much better systems. Back to the motors of Feringa. So here we see the 4 states. This is one of the molecules. So this is what we call the Pp-trans. So P is a nomenclature for helices, for the helix. This is the Pp-trans. So this part here comes towards you. Here you see this part goes to the back of the screen. So here we start. We have a photochemical step. We have this one here flips to the other side, 280 nanometers excitation. So we end up in this state. The MM-cis, we go from the Pp-trans to the MM-cis. Now we need to flip this one relative to the other one. These 2 units have to flip relative to each other. So we have to heat it. We have to heat it to get to this state, Pp-cis, they call it. And then we can have the next light-induced step to get to the MM-trans. This represents the same type of processes as we saw in the movie, where the top of the molecule just rotates around. But it shows there are 2 photochemical steps and there are 2 thermal steps. And the thermal steps, of course, depending on the barrier that you have to go over, you may have to heat it to 80 degrees or something. And you want to lower that barrier. So these are the representations of the molecules of Feringa. Here we start, Pp-trans, we excite it. We get to excited state. We have this double bond isomerization step, and we end up here at the MM-cis. Now from the MM-cis, we have to heat it up. We have to go over the thermal barrier, and we get here, Pp-cis. We have another excitation, photochemical step, and we have to go there. So these barriers, we're overcoming them by using light. Light has a lot of energy. The photons are UV, high energy photons. So the barriers in the ground state, they don't matter. But for the thermal steps, they do. So the final step is again going to the Pp-trans with heating. The first motor molecules of Feringa, they needed really going from the lamp to the oil bath, to the lamp, and again to the oil bath to heat it, to get the full cycle. So of course, he worked a lot on improving the system. He made a molecular car out of it. Here we have the structure, here we have the chassis, and here we have the microscope. So it is scanning tunneling microscope. It has a metal tip. It gives excitation with basically with a current, with electricity. And then you see the movement of the wheels relative to the rod on the surface. So we have isomerization, helix inversion, bond isomerization, and helix inversions. These are the cycles of the molecular motor. And you can not only do that with light, but also with an STM microscope. So this is how they model the movement of this molecular car over the surface. Okay? So you see it's a little bit hopping, a little bit wobbling, it moves. So if the isomerizing groups have the right location, This is how they imagine or how they model with computer modeling how the molecule moves over a surface. So with STM, they've measured the movement of a molecule, this light blob, over the surface. And they have picked different molecules. So it seems this molecule here, it's not the right one. It's making circles. So it's not the right isomer. It's not really going in the right direction. But this one, here you see, this one moves in a line. This is the right molecule. It moves here and it moves, okay, in one direction like you want. So if you put these molecular motors on the chassis, you don't have, 100% control. So some of the molecules have the wrong geometry or the wrong positioning of the motors, and that's why they make circles. But the other ones, they were the right ones, and they move one direction. And this is scanning tunneling microscopy. It also got a Nobel Prize from IBM people. It is atomic resolution. So you can have atomic resolution on a flat surface of molecules. It's a very physical technique. People use it a lot. And we not only have modeling, a nice movie that shows the molecule, but also real physical measurement of the moving of the motors. Feringa has made many molecules, motor molecules, over the years. And this is really maybe a period of 20 or 30 years. And represented here are half-lives of the thermal steps at room temperature. If you don't do extra heating, if you take this one and you wait until it goes to the next state, it takes 1367 years. At room temperature, it takes very long. So they improved the molecule, a better system, 35 hours. A better one, 10 minutes, 15 seconds, 36 milliseconds, up to 8 nanoseconds. So the thermal step at room temperature now takes 8 nanoseconds. So you see the amount of work. This is all synthesis, and it's difficult synthesis. To make a motor molecule can take a good PhD student, maybe even a year. It's a tremendous amount of work, a whole generation of motors that are just getting better and better on the thermal steps especially. Here, another sequence of molecules going from days, hours, minutes, and microseconds. This is a review from Feringa himself. So you can get a feeling of the amount of work, the amount of motor molecules they have produced there in Groningen. It is really amazing. They have also incorporated them in fibers, and in this case, with a muscle-like function. So this is also a Feringa paper using their motor molecules to do something, to make a function. This is a typical representation of a muscle in your body. It has this kind of typical segments that can move relative to each other. It makes you think about maybe Rotaxanes. Stoddart has also worked in this field. So here you see the contraction of a Rotaxane that mimics a muscle type movement in a polymer. But this is what Feringa did. So they used a motor molecule with long alkyl tails, and it can give these different states, the photochemical state and the heated state. What they did, turn it into a fiber and irradiate. So here you see a little thread, and it is irradiated and it moves. The motor molecule, double bond isomerization, can cause physical movement of a macroscopic object. So it is with 365, some heating. Okay, it's a string, it's a little string. And here you see it, this is a human hand. They attach a weight. You have a fiber where the motor is present, and they irradiate it, and it starts moving. So the isomerization of the motor molecules is converted into macroscopic movement with a weight, so it performs work. This is really amazing work, of course. Calculated work, 0.05 microjoule. A big issue in the molecular motor field is how much work can they perform? What can you really do with it? They make a little fiber move, a macroscopic fiber, and this is how they do it. Then you have two states. One state, the photoisomerized state, and it is incorporated into such a fiber, and one of the parts, it expands. So the diameter of the fiber, it changes. The motor molecules, they basically make the fiber expand on one side, and that's why it moves. And you see that the motor molecules have to be packed in the fiber. They have to be like oriented like a micelle. This is all important. And yeah, here you see a representation of the photo actuation. Here you see there's more room here. So it creates extra room, and that's why the fiber expands on one side. The molecular motors of Feringa, they can do work. They can induce effects. It makes me think about plants. Light, we have the movement of a string of a little macroscopic object. What happens in plants? They have regulated growth depending on the lighting condition. So in the plant, there's a sensor. It senses the nature of the light, and it regulates the growth. And so here you see normal conditions, here you see dark conditions. What is happening in the plant? Double bond isomerization. So this is phytochrome, the eye of the plant, and this is the bond that isomerizes. Altogether, it adds up. The Feringa motors that use isomerization, they use an intrinsic process that nature uses in vision, but also the plants use it in regulating the growth. Okay, there's lots of cycles here going through all kinds of intermediate. It is intrinsically the same process. So you have here a double bond and it isomerizes. And that regulates the growth. Why do the other bonds not isomerize as well? Good question. Because the excitation makes the orbitals on that part change. That's what I can say. Here we have the absorption. The plants, they have two forms of this molecule in their leaves. And depending on whether this absorbs the most or that absorbs the most, so the lighting conditions, they regulate the growth. And that is also isomerization, similar to the thread of Feringa that moves with the light. The plants also move with the light. Phytochrome, it's the eye of the plant, and it regulates the growth. And it has a biological cycle just like rhodopsin or similar. These motor molecules, these rotaxanes, what can we use them for? How can we apply them? How can I make a product in which this has a function? That's difficult. But now you have seen an example of this moving thing with light. This is a similar example. It is azobenzene with a long tail on a surface. Depending on the lighting, you can change the hydrophobicity of the surface. So you can, with light gradient, they can move off oil by isomerizing this double bond here with a calixarine on surface with a long alkyl tail. And so you can think about rotaxane systems that change the nature of a surface. That's one thing. Maybe you can make it self-cleaning in that way. Double bond isomerization, that is the important thing. That is the photochemically, it's the step that you use. As a finalization, and we have discussed motor molecules, rotaxanes, and the shuttling. We can do it with electrochemistry, but also with photochemistry. And we can probe the ring, the position, with all kinds of techniques, UV-vis, strange absorption, infrared. But the Feringa motor molecules, it is the internal chirality that's very essential and that makes it unidirectional. In general, isomerization, it can perform all kinds of tasks. Also, holographic material, they're also made with azobenzene polymers. So Cis-Trans isomerization, okay, it's a very useful process